morning, student. Next new concept that is called as cell division as well as cell cycle. Almost all. It is an universal character of all the living organism. All the living organisms, they can undergo cell division, hence they can grow very well. Okay. Then after in the human being, after the formation of an cell, a zygote, then that's a single zygote, it will give rise to what? A multicellular human body. Hence all the organisms, they will origin from what? Single cell. Hence today we want to discuss about cell division as well as or maybe cell cycle or a cell multiplication. We can put as a cell reproduction also. Then, sir, what do you mean by a cell division? Cell division means it is a process of formation of new daughter cell. Process of formation of a new daughter cell from a single parental cell. Here I want to go. Here one cell is there. It can undergo multiplication. It can undergo reproduction. Give rise to what? Two daughter cells are there. Hence, the formation of what? New daughter cell from the, this one is what? Parental cell. Hence, the formation of new daughter cell from the parental cell, that is called as reproduction as well as cell division. Okay. Then within the cell division, then we want to know about the cell cycle. Then within this cell cycle, a cell division it was very well studied by two scientists. Those are called as Boveri as well as Fleming is there. Then within the cell cycle, there are phases are there. Then what you mean by cell cycles, here we want to get what? Two daughter cells from what? Single parental cell. That is called as a cell reproduction. Sir, what do you mean by cell cycles, sir? It is what? It is a period between the formation of new cell or as well as maturation of the cell. That is called as what? Which one? Cell cycle. Sir, what? It is a period between the what? Formation of what? New cell as well as maturation of the cell. Here we want to get the two daughter cells are there. Then this one is here we want to get some periods are there. Parental cell, it can undergo number of phases. Hence the period between, here we want to see what? Some period is there. Here the which one? Period between the formation of what? Two daughter cell, then that period, this period is called a what? Cell cycle. Then we want to see the cell cycle. Within the cell cycle, two phases are there. The first one is called as what? Interphase. Then the second one is what? M phase. First one, we want to see the interphase. That interphase, the cell produces here the cell first it will prepare very well. If we want to attend the examination, first we want to do what? Preparation. Then after the preparation, we are able to succeed in our examination. Hence, in the formation of new daughter cell, first it can go that parental cell, it can go what? Preparation. Then that preparation stage is called what? Interphase. Okay. Then within this interphase, there are three phases are there. Here I will draw the cell cycle is there. Within the interface from this one here, then up to this. Here three phases are there. These are the interface. Okay. Within the interface, first phase is called what? G1 phase or gap 1 phase. Then the second one is S phase. Then the third one is what? G2 phase or gap second phase. Okay. First we want to know about G1 phase or the gap phase. Then sir, one parental cell, it will give us one two daughter cells, sir. Minimum it will require how much time, sir, for the formation of one two daughter cells? Minimum it will require 20 to 24 hours. Yeah. Then we want to get the two daughter cells. First we want to know about the G1 phase. Here I want to draw the I want to show the G1 phase. Within this G1 phase, it is the longest phase within the cell cycle. It will require here one parental cell. It can undergo one G1 phase. Then it can undergo one G2 phase. Then it can undergo one G1 phase, S phase, then G2 phase. Then first this parental cell, it can undergo G1 phase. First we want to know first about S phase. Sir, what happens within this S phase, sir? Within this S phase, first we want to know the S phase. This phase, it is called as what? Synthesis phase. Here, synthesis of DNA will be done. Hence, it is called as what? I think S phase. Here, must and should, I think DNA replication will be done. Hence, I think one set of DNA, it will give us to what? Two sets of DNA, because one parental, I think cell, it having what? 2n number. Yeah. Then here finally we want to get the two daughter cells are there. Then these two daughter cells, they will get how much number of I think a chromosome? 2n number. That is here 46 chromosomes are there. Here these two daughter cells, they will get what? 46. Hence the DNA duplication, it will be done in the S phase. Here, I think DNA replication, it will be done within the S phase. Then this S phase, it will require minimum 8 hours. Then here the centroids will be divided in the animal cell. If the I think DNA, it is I think S phase within the I think animal cell. Here we want to see what centroids will be divided and they will become what? Double. Then the most powerful, I think, DNA packaging. DNA here, some histone proteins are there within the nucleus. Then that DNA, it can wound around histone proteins. Yeah. Then these are the histone proteins are there. Hence, histone proteins will be synthesized within the S phase. Then this is about the S phase. For the purpose of, I think, DNA synthesis, formation of the new daughter DNA molecule within the S phase. First, we want to know what? G1 phase. It will require how much hours? 10 hours. Then here, for the purpose of I think DNA replication, then I think protein synthesis, it will require what? Number of I think enzymes are there. Yeah, DNA helicase is there. Yeah, topoisomerase enzyme is there. That will be required for what? DNA replication. That DNA replication will be done within the S phase. Hence here, I think for the purpose of DNA replication, some enzymes they will be required. Then those enzymes, those will be formed within the G1 phase. Hence, protein synthesis will be done, I think, within this G1 phase. That is, some enzymes which are required for the protein synthesis or the DNA replication, they will synthesize or the manufacture within this G1 phase. Then I think as well as RNA will be synthesized here. Then the adenosine triphosphate, ATP, energy currency of the cell, it will be also manufactured within the G1 phase. Lot amount of ATP will be manufactured within the G1 phase. Then I think raw materials which are required for the manufacture of the DNA molecule within the S phase. Those are what? Nitrogen bases, pentose sugar is there, phosphate group is there. Then those will be manufactured within the G1 phase. If it will prepare very well, those enzymes as well as nitrogen bases, pentose sugar, phosphate group, adenosine triphosphate, they will be synthesized within the G1 phase. It is help for the formation of what? Duplication of what? DNA in the S phase. Okay, this is about I think G1 phase. Then the next one, I think G2 phase of the gap 2. You can put what? Gap 2. It will require minimum 5 hours. Then here once again, I think protein synthesis will be done, especially the proteins which are helpful for the formation of the I think spindle fibers during the cell division. Yeah, then those protein, which one? Tubulin protein is there. That tubulin protein, it will be synthesized within the G2 phase is there. Tubulin protein, it is helpful for what? Formation of the spindle fiber that will be manufactured within the G2 phase of gap 2 phase. Then the plasma membrane proteins, those are also manufactured within the G2 phase. Then the ATP synthesis, it will be increased much more than the I think G1 phase. Then the finally RNA synthesis it will be done. It is most common in the I think G1 phase as well as G2 phase. Protein synthesis will be done. Protein synthesis will be done here, G2 phase. Then RNA synthesis will be done in the G1 phase. RNA synthesis will be done in the G2 phase. These are the most common I think functions of G1 as well as G2 phase. Then this is about the I think interphase. Then the second phase, that is called as M phase. Afterwards, I want to teach it very well. Okay. Then within the M phase, it can undergo. Here I want to mention here once again. It can undergo that parental cell. It can undergo G1 phase, S phase, then G2 phase. Then after preparation, this one is what? Preparatory phase. Then after the preparation, it can undergo what? Mitosis. In the mitosis, we want to get what? Two daughter cells are there. Then that when that prepared, cell, it is ready for undergo what? M phase. That is mitosis. It may be meiosis is there. Then I think first it can undergo what? Which one? This cell, it can undergo, first it can undergo what? Karyokinesis. What do you mean by karyokinesis? Division of what? Nucleus, that is called what? Karyokinesis is there. Then after the karyokinesis, within that karyokinesis, here you will see, after the G2 phase, it can undergo what? This one is karyokinesis is there. Karyokinesis. Within this karyokinesis, you will see what? Within mitosis or it may be meiosis. Prophase is there. Metaphase, anaphase, telophase. Okay, this is what I think karyokinesis. That is the division of nucleus will be done after the preparation of that parental cell. Then finally, it can undergo what? Cytokinesis is there. Which one? Cytokinesis. That is the division of cytoplasm. Division of what? Cytoplasm. Then after the division of cytoplasm, here we want to get what? Two daughter cells are there. Here we want to get what? Two daughter cells. This is phases of cell cycle. Then one more thing here, you will hear the word that is called as G0 phase. Sir, what about the G0 phase, sir? Here I want to mention once again. Here, these two cells, here I want to mention once again. Here, we want to get finally, after the cytokinesis, we want to get what? Two daughter cells. That is called as, cell division or cell reproduction. Then within the G0 phase, as formed after the M phase, two cells, they cannot go into the what? Cell cycle. They cannot go into the cell cycle. These two, these two daughter cells, this one, this one, then they are enter into the G1S and G2 phase. But in the G0 phase, these two daughter cells, they can't undergo what? Cell cycle. What happens, sir? They can't undergo the cell cycle. They will become independent, specialized, function. They will become, I want to do one example. Within the dicot stem, here you will see the epidermis is there. Below this epidermis, you will see the some parenchyma cells are. Which one? Some parenchyma cells are there. These parenchyma cells, here we will get what? Two daughter cells. Then they will become what? Specialized function cell. That is what? Just like a parenchyma, it may be collenchyma. Okay. Then they can do what? Differentiation. What? Differentiation. They can't undergo within the cycle. That is what? Differentiation. That is called what? Differentiation. Once again, those are differentiated parenchyma cells, they can undergo once again to what? Cell cycle. Cell cycle. That is what? Differentiation is there. Differentiation. Then these, here you will get what? Parenchyma cell. When these two daughter cells, they can't undergo the cell cycle, they will become what? Specialized functioning cell. Those are what? Parenchyma cell in the dicot stem. Then these parenchyma cells in the dicot stem, they can undergo what? Differentiation. They will become what? Kbl. Here we want to get what? At the parenchyma cells, they will become what? Kbl cells. Which one? Kbl cells are there. That is, increasing the girth of the stem, it will require in the dicot stem. Okay, those parenchyma cells, they will become what? Kbl. These two Kbl cells, they can undergo G1 phase, S phase, G2 phase, as well as prophase, metaphase, anaphase, telophase. Then cytokinesis. Once again, it will give us to what? Daughter cell. Then these two daughter cells, they can't undergo into the cell cycle. They will become once again specialized type of cells are there. Here, this part Kbl, it will give rise to secondary cortex is there. Secondary cortex. Here we want to get what? that I think Kbl, it can undergo cell division is there. It will give us what? Upper side it will give what? Dead cells, those are what? Cortex cells. Then lower cell, living one, that is what? Secondary cortex. Then they can't undergo, I think, these, I think, which one? They can't undergo the cell cycle, I think. Then once again, sorry, I think these, I think, undergo the cell cycle, give rise to what? I think cortex cell as well as secondary cortex. And this one is what? Which one? Redifferentiation is there. Which one? Redifferentiation. I want to repeat once again. First, here we want to get what? Two daughter cell. They will become what? Specialized functioning cells. Those are what? Parenchyma cells. Then those parenchyma cells, they can undergo the cell cycle. Then once again, I think those parenchyma cells, they can undergo what? Dedifferentiation. They enter within the cell cycle. Yeah, they will give rise to what? New type of cell. That is called what? Kbl cell. Kbl cell. Then these two Kbl cell. Then once again, they can't undergo the cell cycle. They will become what? They will give rise to what? Cortex as well as secondary cortex. This is about the cell cycle. And next class we will discuss another new concept. Thank you.